Hi everyone, welcome to a new episode of the Bioinorganic Chemistry course. Metalloenzymes II. In the last lecture, we have learned about the metalloenzymes, what are metalloenzymes, and in particular, we learned about the zinc-containing enzymes, carboxypeptidase and carbonic anhydrase. In this lecture, we will see other metallobiomolecules and metalloenzymes that are part of biological processes. First, we will discuss about the superoxide dismutase enzyme. What is the function of this enzyme, superoxide dismutase? It is designated as SOD. This enzyme catalyzes the dismutation or partitioning of the superoxide or the O2- radical into either ordinary molecular oxygen or into hydrogen peroxide. So, the function of superoxide dismutase is the dismutation or partitioning of the superoxide radical into molecular oxygen or the less harmful hydrogen peroxide and molecular oxygen. There are different forms of superoxide dismutases. The first one is the copper zinc type, that is, it contains both copper and zinc. That is the first type, copper zinc type copper oxide dismutase. The next type is iron manganese type dismutase. It contains iron or manganese, and there is also nickel type copper dismutase enzyme. So, copper oxide dismutase enzyme usually contains copper. Copper zinc type is there, iron manganese type and nickel type superoxide dismutase are present. The first type of superoxide dismutase can be designated as SOD1 type, that is present in the cytoplasm. The SOD, superoxide dismutase II, is that type of dismutase enzyme that is located in the mitochondria. The superoxide dismutase III type is usually extracellular enzyme. The SOD1 and SOD3, that is superoxide dismutase I type and superoxide dismutase III type, contain copper and zinc, whereas SOD2 contains, it is usually mitochondrial enzyme and it is the manganese containing superoxide dismutase enzyme. Now, let us learn in detail the most important superoxide enzyme, that is the copper zinc dismutase enzyme. It is a metalloenzyme that is composed of two copper and zinc atoms that are bound together by sulfur containing amino acid chains. Here in this figure you can see the copper zinc atom that is bound together by the sulfur containing amino acid side chains. It is the copper zinc dismutase enzyme. The ligands of the copper and zinc are 6-histidine and 1 aspartate side chains. 1 histidine is bound between the two metals. You can see here 1 histidine is bound between the two metals and there is histidine, 6-histidine and 1 aspartame. In the resting state, copper is present in the distorted square planar structure and zinc is present in the tetrahedral structure. In the resting state, we can see that the copper is present in the square planar and zinc is present in the tetrahedral structure. Now, we are moving on to the next category of enzyme, that is the another metalloenzyme, that is the catalase and peroxidase. First, we will see what is the function of catalysis. Catalysis is heme-containing enzyme that will catalyze the breakdown of hydrogen peroxide or superoxide ion into water and oxygen. The catalyze enzyme break down hydrogen peroxide or superoxide ion into water and oxygen. It is usually a tetramer of 4 polypeptide chain. It contains iron in the +3 oxidation state in the high spin form. The 5th and 6th axial positions are occupied by the phenolate aminos of the tyrosyl residue and water molecule respectively. Here you can see the protein structure of the enzyme. In the catalysis will catalyze the breakdown of hydrogen peroxide and superoxide into water and oxygen, that is the less harmful products, water and oxygen. It is a tetramer. You can clearly see the protein structure of the enzyme. It is a tetramer. It is having iron 3 in the high spin state, and the 5th and 6th positions are occupied by the phenolate of the tyrosyl residue, and the other one is occupied by the water molecule. You can see the structure. So, it breaks down hydrogen peroxide into water and oxygen. It contains iron in +3 oxidation state. This is an example of the peroxidase enzyme, that is horseradish peroxidase. You can see that it contains a heme, calcium 2+ and disulfide bonds. Now, let us see in detail what are peroxidase enzyme. Peroxidase enzymes are actually heme containing proteins. Heme containing protein, other examples you know, it is present in hemoglobin, myoglobin, etc. So, peroxide is also a heme containing protein which is found in plants, animals and include human beings also. It is also classified as oxidoreductase and reducing peroxides. An example is horseradish peroxidase C enzyme, which contains 2 types of metal center. We have already seen in the previous slide, that is it contains 1 iron and 2 calcium groups. Here you can see the heme contains iron and it contains 2 calcium group. The peroxide is usually containing iron in the high spin state, and it is surrounded by large protein molecules. We can see here the heme is surrounded by large protein molecules. This is the structure of peroxidase enzyme. Now, we move on to the next category of enzymes, that is the oxidases and oxygenases. As the name suggests, oxidases belong to the class of oxidoreductase and catalyze oxidation reduction reaction using dioxygen as the electron acceptor, leading to the formation of water or hydrogen peroxide as the byproduct. Here you can see the equation. So, it will catalyze the oxidation reduction reaction using dioxygen as the electron acceptor, and it will lead to the formation of water or hydrogen peroxide. For example, of these enzymes are ascorbic acid oxidase, cytochrome C oxidase, monoamine oxidase, xanthine oxidases, etc. This is the structure of the enzyme monoamine oxidases. Now, we move on to the next category of enzyme, that is the cytochrome C oxidase, which is also an oxidase enzyme. It is a very important example. It is the terminal member of the electron transfer chain in mitochondrial respiration. We will learn the cytochrome C oxidase in the electron transport protein lectures in the coming videos. It is the terminal member of the electron transport chain in mitochondrial respiration. It has 4 active metal ions, that is it has 2 copper and 2 heme ions, and it catalytically reduces dioxygen into water. Here you can see that the cytochrome C oxidase enzyme will reduce dioxygen to water. It contains 4 metals, that is it contains 2 copper atom and 2 iron atom. It is also known as complex 4. It is a type of oxidoreductase enzyme in the class of oxidases. Here you can see that the structure of cytochrome C oxidase, it contains 2 heme and 2 copper atoms. The detailed structure will be learnt in the coming lectures. Oxygenases are mainly heme enzymes, which is a subclass of oxidoreductases. It is responsible for breaking down iron, which is distributed all over the body. It also catalyzes the incorporation of oxygen atoms from molecular oxygen to substrates during the oxidation of the substrate. Another example is the cytochrome P450. This is the structure of cytochrome P450, and this is the structure of human tyrosinase enzyme. You should remember that oxygenase enzymes are usually heme containing enzymes. Now, we will see an example, that is the monooxygenase. The tryptophan, pyrrolase and keratinase are example of monooxygenase enzymes. Another category is the dioxygenase enzyme. That means it utilizes 2 oxygen molecules, that is dioxygenase. An example of dioxygenase is phenylalanine hydroxylase and tyrosinase. Of this, the tyrosinase enzyme is quite important because it has an interesting feature, that is, it is a multifunctional copper containing enzyme. It controls the production of melanin and other pigments from tyrosine by oxidation in plants and animals. Another interesting thing to note about tyrosinase is that it is responsible for the browning of damaged fruits and plant tissues. For example, the blackening of peeled or sliced potato exposed to air is caused by this enzyme tyrosinase. So, usually the function of tyrosinase is the controlling the production of melanin and other pigments. That is the main function of tyrosinase. Also, tyrosinase is responsible for the blackening or browning of certain damaged fruits or plant tissues. For example, the blackening of the potato. Now, we move on to the next category of enzyme, that is the nitrogenase enzyme. As the name suggests, we understand that nitrogenase enzyme is responsible for nitrogen fixation, that is the reduction of nitrogen to ammonia. They are produced by certain bacteria such as cyanobacteria and rhizobacteria that resides in the root nodules of certain leguminous plants. There are different types of nitrogenases. Here you can see this is an nitrogenase enzyme which contains FeMO cluster, that is iron molybdenum cluster, and also iron sulfur cluster, etc. The examples, as I already mentioned, includes the molybdenum nitrogenase, we have the iron molybdenum nitrogenase, we have vanadium nitrogenase, we have iron only nitrogenase. The molybdenum nitrogenase enzyme is found in the disotrops such as leguminous plants or rhizobia. The vanadium nitrogenase and iron only nitrogenase is found in Acetobacter. Now, we move on to another category of enzyme, that is the xanthine oxidase. It is found in chicken liver. It contains 2 molybdenum atom. So, the metal present in xanthine oxidase is molybdenum. It also contains 2 FeD molecules and 8 iron atom per molecule. It also contains iron and molybdenum. So, what is the function of this enzyme? It is having the role of oxidizing xanthine into uric acid. The xanthine oxidase enzyme oxidize xanthine into uric acid. It can also catalyze the oxidation of aldehyde to carboxylic acid. It also catalyzes the oxidation of aldehyde to carboxylic acid. Now, let us see another enzyme, that is the aldehyde oxidase enzyme. This enzyme is present in the rabbit liver and it has certain similarities with the xanthine oxidase. Both have molybdenum and iron and FAD, although aldehyde oxidase also contain 2 extra molecules of coenzyme Q. It is present in rabbit liver. It it has certain common features with the xanthine oxidase. It contains molybdenum ion and FAD along with coenzyme Q. So, what is the function of aldehyde oxidase? They utilize molecular oxygen and generate peroxide anion. They generate peroxide anion by utilizing molecular oxygen. They have the ability to catalyze the hydroxylation of aldehyde and N-heterocyclic substrate. Aldehyde oxidase catalyzes the hydroxylation of aldehyde and N-heterocyclic substrate. Now, we move on to the nickel containing enzyme. The first nickel containing enzyme that we are going to learn is the urease enzyme. It is found in jack beans and it contains tightly bound nickel metal which activates the enzyme made up of polypeptide. Urease contains nickel enzyme and it is utilized to catalyze hydrolysis of urea to ammonia and carbonic acid. That is, urease enzyme catalyzes the hydrolysis of urea into ammonia and carbonic acid. Next, we will see a few chromium containing biomolecules. The glucose tolerance factor contains chromium 3 along with glycine, cysteine, glutamic acid, nicotinic acid, and it is thought chromium, to a surprise, it is also thought to be an essential element. Now, we will see some tungsten containing metallobiomolecules. Some of the bacterial enzymes such as formate dehydrogenase contain tungsten. Formate dehydrogenase is known to contain tungsten metal. Another category of enzyme is the phosphatase enzyme. These enzymes contain copper, zinc and magnesium and they act upon organic phosphates to hydrolyze them into free phosphate. What is the function of phosphatase enzyme? In order to convert organic phosphates into free phosphates. The enzyme contains zinc, copper and magnesium metals. Another category of enzyme is the aminopeptidase enzyme. It contains magnesium and manganese and this enzyme hydrolyzes polypeptides at the free amino acid end of the chain and form lower peptides and sometimes free amino acid. As the name suggests, aminopeptidase enzyme hydrolyzes the polypeptide bonds and it will generate free lower peptides and free amino acids. Another category of enzymes are dehydrogenase enzyme. It is an oxidoreductive enzyme. For example, we have alcohol dehydrogenase for alcohol fermentation. Another category of enzyme is the hydrogenase enzyme. Hydrogenase enzymes are usually iron and nickel containing enzymes. So, this hydrogenase enzyme catalyzes the reversible oxidation of molecular hydrogen to protons and electrons by a heterolytic splitting mechanism. You can see here the mechanism. The hydrogenase enzyme will perform the reversible oxidation of molecular hydrogen to protons and electrons by a heterolytic splitting mechanism. Here you can see the structure of several hydrogenase enzymes. Actually, 3 types of hydrogenase enzymes are known. The first one is the nickel iron hydrogenase. We also have iron-iron hydrogenase and also iron hydrogenase. That is iron-iron, 2 iron atoms are present, nickel iron and iron only hydrogenase enzyme. Here you can see the iron only hydrogenase enzyme. This is the 2 iron hydrogenase enzyme and this one is the iron nickel hydrogenase enzyme. Also, we have another category of enzyme known as the isomerases and synthetases. They catalyze the interconversion of optical, geometrical or position isomers. The isomerases or synthetases will catalyze the interconversion of the optical or geometrical or position isomers. For example, we have the vitamin B12 coenzyme. Now we move on to another set of biomolecules, that is the metalloenzymes. In the last few slides, we have seen different types of metallobiomolecules containing different metals and protein chains attached to it. Now we will see in particular metalloenzymes, that is, these are usually the biological catalysts. In the previous slides, we have learnt about certain proteins which are either performing any specific functions. Here we are going to see the biological catalysts. They are known as the metalloenzymes. We know that enzymes, in enzyme structure, we have an apoenzyme part and a cofactor. The cofactor is further containing either a coenzyme or an inorganic cofactor. Here you can see the Hall enzyme, known as the Hall enzyme, is formed by the combination of apoenzyme and cofactor. What is an apoenzyme? It is a high molecular mass that determines the selectivity and rate of the reaction. This cofactor will have a low molecular mass and it will determine the type of reaction. And the hollow enzyme is the fully functional enzyme. Now let us see what are prosthetic groups. Prosthetic groups are actually the cofactors that bind tightly or covalently to the apoenzyme. These cofactors bind very tightly to the apoenzyme. They impart structural property to the enzyme. It is involved in catalytic mechanism and required for its activity. For example, we have the heme group. Heme group as one of the most important prosthetic group. FAD, that is flavin adenosine dinucleotide, several RNA, all these are prosthetic groups available in enzymes. Heme group is the prosthetic group in hemoglobin. Now we will see what are coenzymes. Coenzymes are actually the cofactors which are loosely bound to the transient of the apoenzyme. Prosthetic groups are tightly bound, coenzymes are loosely bound. It defines the functional property of the enzyme. It defines the functional property of the enzyme. For example, NAD and NADP. Then we have the metal ion which loosely bound to the, for example, we have zinc which is bounded to the protein part in carboxypeptidase. So, another important biomolecule or a metalloenzyme is the coenzyme vitamin B12. Coenzyme vitamin B12 is a cobalt containing enzyme with coenzyme B12. We know it is present in vitamin B12. This coenzyme B12 is also found in synthetase, transferase, reductase, mutase and vitamin B12. Vitamin B12 is actually synthesized by bacteria and not found in higher plants. Very important, this vitamin B12 is not present in higher plants, but it is synthesized by certain bacteria and it must be obtained from various food sources. Here you can see the structure of vitamin B12. We will learn in detail in the coming slides. So, now we will see the structure. It is the naturally occurring organometallic complex. It is the first known organometallic complex which is naturally occurring. It is the only vitamin which contains a metal ion. Vitamin B12 is the only vitamin which contains a metal ion. The structure of the molecule is octahedral. It contains cobalt in the +3 oxidation state with a corin ring, a 15-membered 4-nitrogen ring ligand. Here you can see the corin ring system. the cobalt is present in the +3 oxidation state and it is octahedrally geometry. So, why cobalt is present in this particular vitamin? There is some reasons for that. We can say that cobalt can exist in 3 oxidation state, that is cobalt 3, cobalt 2, cobalt 1, and they are stable in aqueous medium and are easily interconvertible from one form to another. Now, let us see the structure of the coenzyme B12 in detail. The side chains attached to the corrin ring are acetamide and propionamide groups with isopropanol phosphate residue attached to the ribose. Here you can see the acetamide, propionamide and the isopropanol phosphate attached to the ribose. In the 5th position, we have 5,6-dimethyl imidazole. Here, 5,6-dimethyl imidazole, it is only present in vitamin B12. No other enzyme has this axial ligand 5,6-dimethyl benzimidazole. The 6th position in vitamin B12 is usually occupied by the cyanide ligand. There are different types of coenzyme B12. Once the R is changed, the name of the coenzyme is also changed. So, you should understand that the basic structure of vitamin B12 contains a corrin ring system with cobalt in the +3 oxidation state, and different types of substitutions are present. The acetamide propionamide groups, isopropanol phosphate residue. In the 5th position, you have, this is an exclusive property of this coenzyme, that is, it is containing 5,6-dimethyl benzimidazole in the 5th position, and the 6th position is usually occupied by cyanide ligand. The active form of vitamin B12 is usually known as methylcobalamin. When the cyanide ligand is being removed, it is called cobalamin. Therefore, the vitamin B12 is called cyanocobalamin. But you should understand that the active form of vitamin B12 is methylcobalamin, but usually it contains the cyanide ligand and it is called cyanocobalamin. Vitamin B12 is called cyanocobalamin, but it is not the active form. The active form is actually methylcobalamin. Here you can see the corrin ring system. The difference is clearly shown in this picture. In porphyrin, you are having the peripheral alternate double bonds. Here you do not have the conjugation or the alternate double bonds, but the basic structure is quite similar. The difference is that this is highly conjugated structure, the conjugation is quite low, this is highly rigid and this is quite flexible. It is having a larger ring size. The hemoglobin that is having the porphyrin ring, we have a larger ring size, and comparatively the ring size is smaller in case of corrin ring system. Most commonly used supplements in cyanocobalamin, that we are taking the supplements of vitamin B12, and when it is converted into active form, it will become, the active form is methylcobalamin. R site is unoccupied usually in vitamin B12, and the 5-coordinated cobalt 1 atom is extremely reactive. Usually the R site of vitamin B12 is unoccupied. Now we will see the different forms of cobalamin, that is different forms of vitamin B12. Here, as I told, the commonly occurring is the cyanocobalamin, that is the vitamin B12 that is synthesized, that is cyanocobalamin, that is the R group is actually cyanide. Now in vitamin B12 alpha, the R is replaced from C into OH. When R is CH3, it is called methyl vitamin B12. The basic structure is same, but different forms as the substituent is differed. We can have 3 different types of vitamin B12, or in various these are the various forms of vitamin B12 when we differ the substituent R. So, in vitamin B12 we should remember that cobalt is present in +3 oxidation state. Because of the spin, I already mentioned that it is present in the octahedral geometric environment. The cobalt 3 in these compounds will have T26Eg0, that is the electronic distribution, and it is usually diamagnetic and EPR inactive. That is, in vitamin B12 it is the cobalt 3 is present in low spin state, diamagnetic and EPR inactive. The red and brown colors of vitamin B12 are caused by the pi to pi star transitions in the Corin ring system. You know that the color of vitamin B12 is a certain brown color, and it is caused by the pi to pi star transition in the Corin ring system. There are different types of vitamin B12. The vitamin B12R has a low cobalt 2 spin. The cobalt 1 in vitamin B12 is also EPR active. Here you can see when the oxidation state of cobalt is 1, it is EPR active and it is having a blue green color due to pi to pi star transitions. And this vitamin B12, which is in this form, that is having a low spin cobalt 2 complex, it is converted into B12A, that is cobalt 3, it is called aquacobalamine, and which on reduction will form B12 cobalt 2, and the B12 or cobalt 2 on further reduction will form B12 as cobalt 1. So, in this module we have learnt about different types of metalloenzymes containing different types of metals, and also we have learnt in detail about the vitamin B12 and the coenzyme B12. Thank you.